Hello everyone. So today I will be talking about bacterial cell structure and its functions. But before that, if there are any noises in the background, please excuse me because that's our neighbor. They're fixing their house and also they're playing loud music. So I can't do anything about that, so please bear with me. So let's start. Bacteria are prokaryotic cells that play an important role in human disease and health. They can cause disease but are also very important in the human microbiota. So right now I will show you something that I created yesterday. This is made up of styrofoam, some acrylic paints, papers, and yeah, that's it. So this is my own representation of a bacterial cell. The outside part is what we call the capsule. The next is the cell wall. Next is the plasma membrane. And then the yellow part, which contains all the parts of the inside, is what we call the cytoplasm. The pink one is what we call the plasmids. Red ones are what we call the food granules. Black ones are the ribosomes. This one, the purple one, is what we call the chromosomes or the nucleoid. The spikes on the outer part of a bacterial cell is what we call the pili, and the tail is what we call the flagella or the flagellum. Okay, so let's start with the outer part of a bacteria. First is the cell wall, which is the blue part. The cell wall is being used by the scientists nowadays to determine whether a bacteria is a gram-positive or a gram-negative. Some bacteria have an extra outer layer part outside their body, which is what we call the capsule. The capsule protects the bacteria and helps in maintaining the shape of the bacteria cell because it encloses the cytoplasm and it defines the cell. It is also the region where processes such as photosynthesis, respiration, synthesis of lipids is occurring. Next is the cytoplasm. As we all know, this is the region where the parts such as ribosomes, plasmids, and food granules are located. Next is the ribosome. This is where the synthesis of proteins occurs. It is where the RNAs are being formulated into proteins. Next is the plasmids. Contains the genes that confer a selective advantage to the bacterium. Next is the chromosomes or the nucleoid, which contains the genetic materials. And next is the food granules, which is of course the part where the bacteria gets its food to consume. Next is the pili, which is used by the bacteria to adhere to surfaces. Next is the flagella or the flagellum. It helps the bacteria to move in different kind of ways, and it moves in circular motion to produce waves that can help the bacteria to move in different directions. That would be all. Thank you for listening.